Okay, so today I'm going to be doing a review on a paper called Monochromatic Blue Light Activates Suprachiasmatic Nucleus Neuronal Activity and Promotes Arousal in Mice Under Sevoflurane Anesthesia. A little bit of background is that monochromatic blue light with a wavelength between 400 and 490 nanometers regulates the non-image forming functions of light in the central nervous system, especially in the SCN. And the SCN is involved in the arousal promoting responses to blue light in mice. Some of the non-image forming functions include what is listed there. The SCN sends both monosynaptic and multisynaptic connections to the hypothalamic nuclei implicated in the sleep-wake cycle. The hypothesis for the study is that it aims to investigate whether monochromatic blue light promotes arousal from sevoflurane anesthesia via activation of the SCN in mice. EEG was used to measure brain activity from the left cerebellum and right frontal cortex. The local field potential was used to record electrical activity measured from the SCN through intracranial electrodes. Ablation of the SCN was administered through electrical lesions of the area. Immunofluorescent staining was used on the lateral hypothalamus, the ventrolateral preoptic area, the SCN, prefrontal cortex, and locus coeruleus to test for concentration of CFOS expression. There were no new methods developed in this study. The EEG and LFP were recorded from the same experimental animal, with or without lesions to the SCN, and immunofluorescent staining happened after the mice went through the emergence from anesthesia, under the effect of either light condition. For the loss of writing reflex, a mouse would be placed on a chamber that would be rotated at 15-second intervals to keep it on its back, and the ability to write itself was assessed. A mouse was considered to have lost the writing reflex if it did not turn itself prone for more than 30 seconds. For the return of writing reflex, it was after exposure to sevoflurane that the mouse was removed from the chamber and placed back into room air. The mouse was considered to have recovered the writing reflex if it turned itself prone onto all 4 feet. The experimental design depicted here was used for the loss of writing reflex and the return of writing reflex. It shows that there are 2 light conditions used in the experiments, polychromatic white light and monochromatic blue light. The loss of writing reflex marks the induction of anesthesia, whereas the return of writing reflex marked the emergence from anesthesia. In Figure C, you can see that the emergent time in monochromatic blue light conditions showed no significant change, while Figure D showed that when induced by anesthesia, the emergence time in blue light conditions significantly decreased, while having no effect of the emergence time in polychromatic white. The researchers analyzed cortical EEG activity for a duration of 100 seconds after stopping the anesthesia. They found a faster transition from an anesthesia state to a wake state after the blue light treatment compared to the white light group during the sevoflurane emergence period. Figures 1G and H indicate that blue light accelerated cortical arousal during the sevoflurane emergence period, and these findings demonstrate that blue light can promote behavioral arousal from sevoflurane anesthesia and increase cortical arousal during the emergence period. After the mice were under anesthesia for 30 minutes in room light in cylindrical glass anesthesia chambers, they continued to be anesthetized but at the same time were administered either blue light or white light at the same illumination intensity. The results showed that the burst suppression ratio response during continuous inhalation under blue light decreased the burst suppression ratio, while the white light did not. The mice that were treated with white light showed no significant difference throughout the 30-minute experiment. However, Figure 3D shows that the mice showed a significant decrease of 31% during the first 10 minutes after blue light. 10 minutes later, the decrease was 42% after blue light, and the last 10 minutes showed a 55% decrease post-blue light. Administration of blue light changed the burst suppression pattern to more burst occurrences. The EEG power after the blue light administration was higher than before the blue light. The combined power spectral densities calculated before and after blue light administration showed that the power at all of the frequency bands except for the spindle band significantly increased. This shows that blue light could increase cortical activation during continuous deep anesthesia. The white light treated animals again showed no statistically significant difference in EEG power compared to white light. Blue light exposure under sevoflurane anesthesia caused a significant increase in CFOS expression in the SCN. This data suggests that the blue light exposure under sevoflurane could enhance SCN neuronal activity. The SCN local field potential activity significantly increased after the application of blue light compared to before blue light. Figure D is a schematic of the experiment. EEG and local field potentials were simultaneously recorded, and the white light treated animals showed no statistically significant difference in the SCN local field potential power compared to before the white light. The blue light treated animals increased the SCN local field potential power compared to before blue light administration. The local field potential in a mouse before white light and after white light showed no significant change, while the local field potential amplitude decreased and the frequency increased after the application of blue light. The ablation of the SCN did not alter the induction time between the white light and blue light groups, but the difference in the emergence time between white light and blue light in sham mice disappeared when the SCN neurons were ablated. These results suggest that the SCN is a modulator for the arousal-promoting effect of blue light under sevoflurane anesthesia. In the SCN lesion group, there were no differences in sham mice in EEG traces, power spectrogram, and normalized group PSD. The application of blue light in the SCN ablated mice did not cause a wake-promoting response in behavioral tests and EEG recordings. This shows that lesions in the SCN reverse the behavioral and cortical arousal from sevoflurane anesthesia induced by blue light. In the sham group, the application of blue light induced a significant 21% decrease in the burst suppression ratio during the first 10 minutes, a 39% decrease during the second 10 minutes, and a 56% decrease during the last 10 minutes after blue light, compared to before blue light. The application of polychromatic white light showed no significant difference in the burst suppression ratio compared to before the white light. The SCN lesions reversed the decrease in the burst suppression ratio induced by the blue light. For the SCN ablated mice, the application of the blue light had no significant impact on the ratio compared to before the application of blue light, whereas lesions in the SCN neurons did not affect the burst suppression ratio between before white light and after white light groups. In the sham group, the power at all frequency bands significantly increased after blue light exposure, while the mice treated with white light showed no statistically significant difference in EEG power compared to pre-white light. In the SCN lesion group, blue light administration did not cause EEG changes in EEG traces, and the SCN lesions did not affect the EEG power between white light pre and post groups. These results show that the SCN lesions reversed the decrease in burst suppression ratio induced by blue light. The application of exposing mice to the blue light showed no statistically significant difference in CFOS expression in sleep-promoting nuclei of the ventrolateral preoptic areas. Additionally, the CFOS expression in the locus coeruleus was not significantly different in blue light and compared to the white light groups. The blue light application did cause a significant increase in CFOS expression in the lateral hypothalamus orexigenic neurons. Here, the data shows that CFOS expression in the prefrontal cortex significantly increased after blue light application when compared next to the white light group. These results are consistent with the previous EEG results that blue light could increase cortical EEG activity under sevoflurane anesthesia. This study found that blue light shortened emergence time but had no effect on the induction time. This suggests that blue light administration leads to faster emergence from sevoflurane anesthesia. The EEG results showed that blue light decreased the relative EEG delta power and increased the spindle beta delta power and increased the spindle beta low gamma and high gamma power, showing that blue light accelerates cortical arousal during the sevoflurane emergence period. This further confirms that blue light can produce arousal promoting effects during anesthesia. The study also found that the SCN lesions got rid of the arousal promoting effects of the blue light and reversed blue light induced decreases in the burst suppression ratio. This finding implies that the SCN plays a key role in regulating the arousal promoting effects of blue light under anesthesia. There are currently no approved interventions that can be used routinely to promote emergence from general anesthesia. It is known that bright blue light influences many things like immunity, inflammation reduction, and enhanced bacterial clearance. Blue light excites lateral hypothalamic neurons under sevoflurane anesthesia, so using environmental blue light during anesthesia recovery may promote emergence and strengthen analgesia, which is worth looking into for further studies. Immunofluorescence staining showed that the application of blue light under sevoflurane anesthesia increased CFOS expression, but a further study using specific neuron conformation is needed. SCN LFP recordings and bilateral SCN electrical lesions are very tough techniques, and more selective methods are needed to further analyze the function of SCN-specific neurons. Also, SCN electrical lesions could possibly destroy projections from the retina to other brain areas. Therefore, they need to observe whether or not neuronal projections from the retina are intact in SCN lesion mice. They also did not examine the effect of SCN lesions on the blue light-induced changes in CFOS expression in the lateral hypothalamus and prefrontal cortex, so further studies can also be done on that as well. This study focused only on male mice, which may make the results unrepresentative of the general species. The relevance of this result in a clinical setting remains to be determined because the experiments were done on mice and not diurnal animals. Thank you for listening to my presentation. I'm sorry for all the technical difficulties, but I hope you enjoyed.